TRIP12 gene. And the long name for the TRIP12 gene is called the thyroid hormone receptor interactor 12, which encodes the E3 ligase in the ubiquitin pathway. This has been associated with the autism spectrum disorder. In addition to autistic features, the TRIP12 mutation carriers show moderate to severe intellectual disability. More recently, TRIP12 was postulated as a novel candidate gene for intellectual disability in a meta-analysis of published ID cohorts. It is not surprising that many members of the ubiquitin pathway have been associated with diseases including cancer such as BRCA1, the associated breast and ovarian cancer, neurodegenerative diseases such as Parkinson and Alzheimer's disease, neurodevelopmental phenotypes, and autism spectrum disorder. Hence, mutations in members of the ubiquitin pathway, and especially E3 ubiquitin ligases, are important contributors to the neurodevelopment pathophysiology. The identification of novel genetic causes of intellectual disabilities and autism spectrum disorder have been the subject of intense research. First thing, we will jump into something called the ubiquitin pathway. The ubiquitin pathway is an enzymatic cascade including activating E1, conjugating E2, and ligating E3 enzymes, which govern protein degradation and sorting. It is crucial for many psychological processes. Compromised function of members of this ubiquitin pathway leads to a wide range of human diseases, such as cancer, neurodegenerative diseases, and neurodevelopmental disorders. For cells to work properly, there must be a healthy balance of unwanted proteins being broken down and new proteins being made from them. This recycling process is necessary to prevent damage or surplus proteins from building up and becoming toxic to the cell. It relies mainly on structures known as proteasomes. Any unwanted proteins attacked with a chain of proteins called ubiquitin. Any protein with a ubiquitin tail means that it's destined for a proteasome. Proteasomes are shaped like barrels. They open at one end to allow tagged proteins to enter, and at the other end allow broken down pieces of the protein to exit only. Tagged proteins are captured by proteasomes. Once captured, the protein unwinds and enters the proteasome inside. The broken down pieces are then released and recycled to make new proteins. Sometimes proteasomes can be overwhelmed when cells generate large amounts of unwanted proteins. When this happens, a different clearing system is used. Instead, tagged proteins aggregate into structures called egg reserves. These are cleared by other structures called lysosomes, which get rid of the proteins instead of recycling them. As I've learned more about the TRIP12 gene and Isabella's missing exons 37 through 41, her partial deletion, it's been pretty overwhelming. From the surface, individuals with TRIP12 deletions or mutations can have developmental delay, extremely short stature, small hands, dysmorphic facial features, hearing loss, and epilepsy. But there's a lot more at stake due to the transcription factors and eventually what could be the lack of cell degradation. The predicted 1992 amino acid TRIP12 protein contains a putative transmembrane domain. Analysis detected TRIP12 expression in all 16 human tissues, with the highest expression in skeletal muscle and testes, lower expression in the heart, kidney, spleen, thymus, prostate, ovaries, placenta, and white blood cells, and other low expressions in the brain, lung, liver, pancreas, small intestine, and colon. The thyroid hormone receptors are hormone-dependent transcription factors that regulate expression of a variety of special target genes. They must specifically interact with a number of proteins as they progress from their initial translation and nuclear translocation to heterodimerization with retinoid X receptors, functional interactions with other transcription factors, and the basic transcriptional apparatus, and eventually degradation. It's interesting to learn the protein expression of this gene of Isabella's exons 37 through 41. They interact with the following with a median read count per base of 3.2 to 9.9, testes, cells, cultured fibroblasts, liver, kidney, whole blood, spleen, pancreas, brain, frontal cortex, kidney cortex, heart, left ventricle, brain, spinal cord, brain cortex, brain cerebellum, brain cerebellar hemisphere, So in summary, the TRIP12 gene is part of the HECT domain E3 ubiquitin family, and the TRIP12 protein is expressed in multiple tissues and thought to play an important role in a number of processes, including ubiquitin fusion degradation and regulating DNA repair and cell growth. So as a result of this gene deletion and others that have the gene mutation of the TRIP12 gene, there's definitely a strong presence of neurodevelopmental issues. There's developmental delays, speech delays. Some children are nonverbal. Many have autistic behaviors. Many have, hypertonia, seizures, vision problems, hearing problems, OCD, ADHD. You can imagine that when your cells, you have 2 billion cells per day that are recycled, where they're taking out the trash and or they're being completely killed off. 2 billion per day, and this gene is essential to that function, and we still don't know to the extent of the impact that it's having on Isabella. That's something that we need to do more research, and we need to find answers. So answers are what we're going after. Presently there is no cure for Isabella. There's not a lot of research on the TRIP12 gene, and that needs to change. This video is part of that call for resources, for researchers that want to learn more about the TRIP12 gene and want to help individuals like Isabella that have a deletion or a mutation find some treatments and treatments that can help her.